Human genetic engineering can be used to treat diseases, to acquire desired traits, and even to treat blindness. Although there can be positive attributes to human genetic engineering, there can also be negative attributes, such as manipulating baby genes to acquire a perfect baby with desired traits, also known as designer babies. Optogenetics is a biological technique which involves light to control cells in living tissue, typically neurons, that have been genetically modified to express light sensitive ion channels. The ultimate goal is to modify the neutrons so that they will be responsive to light. Retinitis pigmentosa is a chronic hereditary eye disease characterized by black pigmentation and gradual degeneration of the retina. Retinitis pigmentosa causes cells in the retina to degenerate, eventually causing people to go blind. Doctors will inject a harmless virus loaded with DNA from photoreceptive algae into the eyes of 15 patients suffering from retinitis pigmentosa. A virus containing DNA will coax ganglion cells will produce a light-sensitive protein called channel rhodopsin, the same protein used from algae to detect light. Doctors will attempt to transfer the duties of photoreceptor cells to different cells in the eyes to restore light. Previous attempts to restore vision using optogenetics in mice and monkeys were successful. Optogenetic approaches, in which neurons express light-activated ion channels so that their activity can be controlled with light, therefore created a great deal of excitement. Optogenetics can target specific genetically defined groups of neurons, allowing a much finer level of control of their activity than was previously possible. Genetic engineering is used to add a channel rhodopsin protein found in algae in order to detect light so that the person will be responsive to light. Light stimulation could be a potential benefit for neurological diseases using gene therapy. Gene therapy with optogenetics has been successful for the cure of Leber's congenital amaurosis by replacing the missing retinal isomerase. Recovery of vision could be obtained with the retina of the animal's regained photosensitivity, which is transmitted by the optic nerve to the brain. Trajectories of the movement of the animals in the dark and in the light show clearly an increased activity in the light as it is obtained for wild-type animals. It is conceivable that such an approach might be possible for blind humans. Some potential drawbacks are when the light-sensitive ganglion cells begin firing, the brain won't recognize the signals. Channel rhodopsin protein only responds to intensive single-colored light, like blue light. This light has to be very bright to trigger the light-sensitive molecules. Limited success is shown through the artificial circuits connected to the neurons in the eye to restore sight. This article mentions the process of optogenetics, which begins by injecting a virus with channel rhodopsin, a special protein used to detect light. A chain of cells in our eyes convert light into electrical signals. In this chain, photoreceptor cells in the retina are reactive to photons, particles of visible light. Retinitis pigmentosa causes these cells to degenerate, eventually resulting in blindness. The goal is to restore vision by creating more ganglion cells, which sends signals from the retina to the brain, causing the eye to be sensitive to light. If optogenetic therapy is possible in humans, there would be no need for surgery or cable implants used in neuroscience, resulting in a lower risk of complications. As previous tests have shown, optogenetics can restore vision in mice and monkeys, so there is a greater possibility of optogenetics restoring sight in humans. But this brings some drawbacks as well. For example, the brain might not recognize the ganglion cell signals. The channel rhodopsin protein detects intensive blue single-colored light. Although there are some disadvantages, there are also advantages. Stem cells is a possible solution to blindness. As researchers discover more about stem cells, it could be possible to put a stop to the progression of degeneration and even repair cells in the human eye. The intended audience is those suffering from retinitis pigmentosa because the article mainly focuses on that particular disorder, which eventually leads to blindness. The intended audience could also be people interested in learning about the process of optogenetics in comparison to neuroscience, implanting fiber optic cables, or surgery. This article focuses more on the advantages and the possibility of optogenetics used in restoring vision. Although not completely discarding the disadvantages, the article mentions very few disadvantages in comparison to the advantages through the explanation of the process. It mentions that the process is less harmful than other processes, taking bias in optogenetics. Although there are minor problems, the possible solution stands out throughout this article. Stem cells could represent the solution to blindness. Optogenetics could be the cure for blindness. But we don't have enough information on the disadvantages. A possible solution for optogenetics is further research and possibly human testing, since testing in animals seem to work. The biggest problem with optogenetics is not knowing the harms it could cause. The largest concern regarding optogenetics is if it could potentially have irreversible consequences. Optogenetics could alter brain activity. The article focuses on the advantages and disregards potential human side effects as compared to animals. There are more possible harmful side effects found in opposing views. I believe optogenetics has a high potential to overcome blindness because it has been tested on animals and been proven to work. Although we are humans, it still leaves a greater potential for the process to be successful. I think the side effects should be looked at further to prevent any potential harm optogenetics may cause in our brain. The article's overall purpose is to explain the potential that optogenetics has to cure blindness and resolve many other problems. The article displays important information regarding the advantages in the process of adding a light-sensitive protein to assure that they will be able to see light found in the algae, which is supposed to be injected with a harmless virus. This is helpful in getting closer to curing blindness. Could optogenetics be the cure for blindness? Although it seems to have a higher possibility of curing blindness because it has been proven in animals, it may still have too many non-proven side effects which could be harmful. Could optogenetics be harmful to the brain and the brain activity? Possibly it could be harmful because a light-sensitive protein is added into the eye to make it receptive to a bright light. The sensitivity to light generated by this protein could have harmful effects whenever they see bright lights. Could optogenetics have long-term side effects? Although not yet proven, it could have potentially harmful risks, which we are not ready to face. It might even hinder our brain functions.